Good morning, everyone. It's a privilege to address such a distinguished group of researchers today as we delve into a critical area of gerontology, the biology of aging. Our focus for this session is on oxidative damage, a fundamental process that underpins many aspects of age-related decline and disease. This discussion is part of a broader series dedicated to providing comprehensive insights into the science of aging, brought to you by the American Federation for Aging Research, or AFAR. For decades, AFAR has been at the forefront of supporting innovative research that seeks to understand the mechanisms of aging and develop strategies to promote healthy longevity. Our aim today is to provide a foundational understanding of oxidative damage, setting the stage for deeper exploration into its implications and potential interventions. The concept of aging is multifaceted, encompassing a complex interplay of genetic, environmental, and lifestyle factors. Within this intricate web, cellular and molecular damage accumulates over time, contributing to the progressive loss of physiological integrity and increased vulnerability to disease. Oxidative damage stands out as a particularly pervasive and well-studied contributor to this age-related deterioration. It represents a continuous challenge to cellular homeostasis, impacting virtually every biological system. Understanding the origins, mechanisms, and consequences of oxidative damage is not merely an academic exercise. It is essential for developing effective strategies to mitigate its effects and extend health span. As researchers, our collective goal is to unravel these complex biological processes. By dissecting the role of oxidative damage, we can identify novel therapeutic targets and preventive measures. This introductory guide aims to synthesize current knowledge, highlighting key concepts and providing a framework for further investigation. We will explore how reactive oxygen species, or ROS, are generated, how they interact with vital cellular components, and the downstream effects that manifest as hallmarks of aging. The insights we gain from this foundational understanding are crucial for advancing our field and ultimately improving human health. We encourage you to consider the implications of this information for your own research endeavors, whether you are focused on basic science, translational studies, or clinical applications. The journey into the biology of aging is a continuous one, and oxidative damage remains a central pillar of this exploration. Let's now define what we mean by oxidative damage and explore its profound impact on the aging process. At its core, oxidative damage refers to the detrimental effects caused by reactive oxygen species, or ROS, on the fundamental building blocks of our cells: fats, proteins, and DNA. ROS are highly reactive molecules containing oxygen, formed as natural byproducts of normal cellular metabolism, particularly during mitochondrial respiration. While some level of ROS is essential for cellular signaling and immune function, an imbalance between ROS production and the cell's antioxidant defense mechanisms leads to a state known as oxidative stress. It is this oxidative stress that precipitates the damage we are discussing. Consider the ubiquity of this process. Oxidative damage occurs in every single cell in the body, continuously challenging cellular integrity. When ROS interact with lipids, they can initiate lipid peroxidation, compromising the structural integrity and function of cell membranes. This can lead to increased permeability, impaired transport across membranes, and a cascade of inflammatory responses. Similarly, proteins are highly susceptible to oxidative modification, which can alter their structure, reduce their enzymatic activity, and impair their ability to fold correctly. Damaged proteins can aggregate, forming insoluble clumps that interfere with cellular processes, a phenomenon observed in various neurodegenerative conditions. Perhaps most critically, ROS can inflict significant injury to genetic material, specifically DNA. Oxidative DNA damage can manifest as base modifications, strand breaks, and cross-links, all of which can lead to mutations. If these mutations occur in critical genes, they can disrupt cellular function, impair DNA repair mechanisms, and contribute to genomic instability, a recognized hallmark of aging and a driver of carcinogenesis. The accumulation of such damage over a lifetime is a major factor in the age-related decline of cellular function and tissue repair capacity. The implications of this widespread cellular damage are far-reaching. Research has increasingly implicated ROS and oxidative damage as critical players in the development and progression of numerous age-associated diseases. This includes prevalent conditions such as cancer, where oxidative DNA damage can drive oncogenic mutations, heart disease, where oxidative stress contributes to endothelial dysfunction and atherosclerosis, diabetes, linked to oxidative damage in pancreatic beta cells and insulin resistance, and neurodegenerative disorders like Alzheimer's disease, where oxidative stress is a prominent feature in neuronal pathology. Understanding these intricate connections is vital for developing targeted interventions to combat these debilitating conditions and promote healthy aging. Building on our understanding of oxidative damage, let's now consider its specific manifestations in certain age-related conditions and explore some of the popular antioxidant compounds that have been investigated for their potential protective roles. Oxidative stress is not merely a general contributor to aging. It plays distinct and significant roles in the pathophysiology of specific diseases. For instance, in Parkinson's disease, a neurodegenerative disorder characterized by the loss of dopaminergic neurons, oxidative stress is a prominent feature. The substantia nigra, the brain region most affected, is particularly vulnerable due to its high metabolic activity and dopamine metabolism, which can generate ROS. Mitochondrial dysfunction and impaired antioxidant defenses further exacerbate oxidative damage in this context, contributing to neuronal death. Similarly, in age-related hearing loss or presbycusis and certain forms of deafness, oxidative damage to the delicate hair cells and neurons of the inner ear is a significant factor. These cells are highly metabolically active and susceptible to ROS-induced damage, which can lead to their degeneration and subsequent hearing impairment. Macular degeneration, particularly the age-related form, is another condition where oxidative stress is strongly implicated. The retina, especially the macula, is exposed to high levels of light and oxygen, making it prone to oxidative damage. This damage can lead to the accumulation of cellular debris, inflammation, and the degeneration of photoreceptor cells, resulting in central vision loss. Given the pervasive nature of oxidative damage, considerable research has focused on the role of antioxidants, compounds that can neutralize ROS and mitigate their harmful effects. Among the most widely studied are vitamin C and E. Vitamin C, a water-soluble antioxidant, directly scavenges free radicals and regenerates other antioxidants. Vitamin E, a fat-soluble antioxidant, protects cell membranes from lipid peroxidation. Carotenoids, such as beta-carotene, lutein, and zeaxanthin, are potent lipid-soluble antioxidants found in plants, particularly important for eye health. Selenium, an essential trace element, is a crucial component of antioxidant enzymes like glutathione peroxidase. Coenzyme Q10, or CoQ10, is another vital compound acting as an antioxidant and playing a critical role in mitochondrial energy production. Its levels tend to decline with age, prompting interest in supplementation. Finally, synthetic antioxidants like BHT and BHA, commonly used as food preservatives, have also been studied for their antioxidant properties, though their long-term health effects in humans are still a subject of ongoing research. The investigation into these and other antioxidant compounds continues to be a vibrant area of research, seeking to understand their precise mechanisms of action, optimal dosages, and efficacy in preventing or treating age-related diseases. Continuing our exploration of antioxidants, it's clear that these compounds represent a diverse group with varied mechanisms and potential health benefits. Beyond the well-known vitamins and minerals, other naturally occurring compounds like melatonin and flavonoids also exhibit significant antioxidant properties. Melatonin, primarily known for its role in regulating sleep-wake cycles, is also a potent direct scavenger of free radicals and can stimulate antioxidant enzymes. Its ability to cross biological barriers, including the blood-brain barrier, makes it particularly interesting for protecting neural tissues from oxidative damage. Flavonoids, a large class of plant compounds found in fruits, vegetables, and tea, possess strong antioxidant and anti-inflammatory activities. Different subclasses of flavonoids, such as anthocyanins, catechins, and quercetin, have been linked to reduced risk of various chronic diseases, partly due to their ability to neutralize ROS and modulate cellular signaling pathways. Beta-carotene, a precursor to vitamin A and a prominent carotenoid, has been extensively studied for its antioxidant capacity. While observational studies have often shown an association between higher dietary intake of beta-carotene and a lower risk of certain diseases, intervention trials have yielded mixed results, highlighting the complexity of antioxidant supplementation and the importance of context. The interplay between different antioxidants, their bioavailability, and individual genetic variations can all influence their effectiveness. This underscores the need for continued rigorous research to fully elucidate the role of specific antioxidants in human health and disease prevention. The broader implications of antioxidant research extend to understanding how dietary patterns and lifestyle choices can influence our endogenous antioxidant defenses and overall oxidative stress burden. A diet rich in a variety of plant-based foods naturally provides a spectrum of antioxidants and other beneficial phytochemicals that work synergistically. This holistic approach to nutrition is often more effective than relying on isolated supplements. The ongoing challenge for researchers is to move beyond simple correlations and establish causal links, identify optimal intake levels, and understand how antioxidants interact within the complex biological systems of the human body. As we conclude our discussion on oxidative damage and the role of antioxidants, it's important to reiterate the American Federation for Aging Research's commitment to advancing this vital field. AFAR continues to support groundbreaking research that explores the fundamental mechanisms of aging, including oxidative stress, and seeks to translate these discoveries into practical strategies for healthy longevity. We encourage you to engage with AFAR's resources and initiatives, which are designed to foster collaboration and accelerate progress in aging research. You can find more information about their work, publications, and funding opportunities by visiting their website at www.afar.org and connecting with them through their social media channels. Your continued dedication to research is paramount in our collective quest to understand and ultimately mitigate the impact of aging on human health.